And without further ado, I'd like to invite our first speaker for the session, Jonny Gershomi, who will talk about the title presented here.
Thank you very much. I need to put things into context. Over the last session, there's been impressive, overwhelming data proving that vaccines work. Now, vaccines can only work if people get vaccinated. And this is a major, major problem. In this current audience, we're all used to analyzing numbers, comparing data, and convinced that this is an amazing accomplishment, the vaccines, which they are. However, the vast majority of the population is confused. And many of the population, due to the confusion and due to the numbers and due to the fact that scientists proclaim that vaccinations work, they're convinced that this is all a conspiracy and I'll be the last person to get vaccinated. So we have a problem, a serious problem. Now, I've been studying viruses, the immune response, and vaccines for many years, and so presumably I should know something about this topic. However, I decided to actually call my lecture Vaccine Lessons I've Learned Recently. It's turned out that because of Corona, almost everything I thought I knew 2 years ago has turned out to be somewhat incorrect. I've certainly learned a lot of new messages, and it's very difficult to convince others of the general population that vaccines are really absolutely necessary when everybody is confused and saying that we're getting vaccinated once, twice, 3 times. Who has ever been vaccinated 4 times in a year? And then confronted with the reality that we see that everybody who's been I have got 10 grandchildren. They've all been double and triple vaccinated, and they've all come down with COVID. They've all recovered, and that's our reality. So what's going on? Now, another thing is we need a lesson here of humility. We were the first country in the world to reach 50% coverage. And so all of our leadership told the whole world that everybody's looking at Israel, look at how much we've accomplished. We are winning, and this is not a competition. And the reality is, if you look today in world data sites, we're listed as ranked 75 in the vaccine coverage. We are around 66% of fully vaccinated, where you have other countries such as Portugal and many, many others, Iceland and France and others, that are much more covered than us. So we have to consider about what have we learned, and more importantly, how do we educate the general public to embrace vaccines for the future, because that's the bottom line of what I would like to say. Now, the first premise was that herd immunity is the answer to the pandemic. Most people 2 years ago didn't know what herd immunity was, but everybody convinced them that this is what we really need. And there are a lot of parameters in order to reach herd immunity. So we need a percentage of the population immune either by natural courses or by vaccination. But there have been various changes in the dynamics of the epidemic which require us to constantly change our perception of how to reach herd immunity. Now, one of the things that was really difficult was the Great Barrington Declaration in October 2020, where basically what is being said is a bunch of public health people and epidemiologists said that we want to change the whole we need to reach herd immunity, but let's do it naturally. Let's let the virus take its natural course. So you have England and Sweden as the model. We don't have to lock down everything. Younger people basically should just go about their lives, get infected. It's not a big issue. And what we really want to do is lock down old people like me. So basically what was said is we have to have focused protection. We're going to protect the elderly here, and otherwise, life will just go on. Now, clearly, this has not been very successful, and there have been a number of issues that this fundamental concept, which was presented in Great Barrington, Massachusetts at the time, was that, first of all, it's really impractical to isolate and lock down the elderly. A lot of us are still very active. We have kids, grandkids. We want to go out to the mall. We want to see a movie. And those who are in the elderly situation that they're in homes, then the staff is going to take care of them today and then go home in the evening and get infected and bring it. So you cannot, it's impractical to selectively lock down a specific segment of the population. The other thing is that the fatality rate ultimately was too high. If you basically say we're going to let the virus take its course and people are going to get infected, it turns out that here today in Israel, there are over 10,000 people who have died. There are over 6 million people who have already died in the pandemic worldwide. And if you calculate what those numbers would have been without the lockdowns and without the vaccination, it would be enormous. So that's impossible. Now, it also turns out, a harsh reality, that some young people die. So it isn't restricted exclusively to above 65. And more than that, we realize that there are ramifications of the infection, such as PIMS and long COVID, which were not anticipated in the original formulation of let the virus take its course. Now, of course, we learned a few other things, such as our immunity is not robust, sterilizing, and long-lasting. And we are confronted with the fact that there is a rapid waning within months. And we see that even the naturally infected, I'm not talking about the vaccinated, the naturally infected get reinfected. So the formula, the concept of here, all you need to do is get infected once and you'll have robust sterilizing immunity for the rest of your life is simply not true. And then, of course, the last parameter here is that in contrast to what many of us said, including me, coronavirus with a huge genome and the ability to repair the RNA and proofreading is not going to be highly variant. And we were wrong. We realized that constantly, serially, we're seeing an increase in mutations as well as new variants that have been selected for super infectious transmissibility. And that would require higher levels of protection in order to achieve the herd immunity. So it is clear that vaccination is absolutely necessary in order to get sufficient coverage and protection within the society. Now, for this, we had a revolutionary development in vaccines, and that is the transition from the traditional hardware vaccines where protein, either as live attenuated, killed vaccine, or subunit vaccine, have been translated into what I refer to as software vaccines, in which we actually use either RNA or viral adeno DNA cloned blueprint for the spike protein, which would be expressed in the vaccinee as opposed to manufacturing. And this was generally a breakthrough, and in fact it was. And if we look at how did we come by that, one of the objections of the public is that, well, you know, it's been 1 year. Obviously, this new technology has evolved within a year in reaction to the pandemic, and therefore it's certainly not safe. And we heard in the last lecture the perceptions of what constitutes safeness. And this is the major problem. We have to convince not ourselves, 1,000 scientists sitting around in a room looking at data and saying, of course I'll get vaccinated. Not all of them are vaccinated, but many are. The problem is convincing the public. So I want to mention the fact that there were 3 innovations that brought the messenger RNA vaccine to be. The first was it had to be realized that the spike protein can assume 2 configurations, a pre-fusion and a post-fusion. And Barney Graham, who was studying RSV and then HIV and MERS coronavirus, realized that by introducing 2 proline mutations, the heptad repeats. This can stabilize the spike protein and make it a very effective and efficient immunogen. This was about 10 to 15 years of research that brought the realization that the minute in January 10th, 11th, where the genome of CoV-2 was published, it was immediately translated into a stabilized spike. And if you ask yourself, for example, what is the difference between AstraZeneca and J&J vaccines? AstraZeneca do not use the stabilized spike, whereas J&J does. And so we see variations in the efficacy. The second, of course, was the use of messenger RNA. Now, this too is not a new technology. I remember at the end of the 80s, people were talking about introducing gene therapy, and therefore we're saying we have to be able to inject DNA or RNA. The major problem with RNA is that it elicits a robust and violent innate immune response, and due to Katie Carrico on the one hand and Drew Weissman, her collaborator at UPenn, they realized that the introduction of pseudouracil as opposed to uracil will make the RNA compatible and therefore could be used. And the last is, of course, lipid nanoparticles, in which here we can mention Peter Cooley's in Canada, who came out with the right formulation to package the RNA and use it. And this has been an amazing, amazing accomplishment and found to be extremely useful. So the question is, however, obviously, according to all the fake news and all the publication, RNA is bad. RNA is really dangerous, and therefore this is the first time we're using RNA as the active ingredient of a vaccine, so it certainly must be dangerous. So let's stop and think about how dangerous is RNA in a vaccine? Well, in reality, you have to ask, is this novel? And again, in reality, we have to realize that there are 7 vaccines given to our babies in Kupat Cholim, in Tipat Chalav, which all contain live attenuated RNA viruses. So when we inject any one of the following vaccines, not meaning that flu is necessarily gray as opposed to hepatitis, which is black hen, all that we are saying is that when you inject a live attenuated RNA virus, it is infecting our cells, it is replicating, generating massive amounts of RNA to go on and infect further for weeks and sometimes months. And when we take the RNA, 10 or 30 micrograms of synthetic RNA from Pfizer or Moderna respectively, and inject it, and it's lost within a few days, if not hours, obviously we have been using RNA in our vaccines for many, many years. So we have to ask ourselves, what considerations should we consider when we talk about the safety of using RNA vaccines such as the ones used now? So first of all, I just mentioned, the RNA live attenuated viruses used for vaccines have been used for over 50 years. Billions and billions of kids have been vaccinated, and there are no indications of long-term side effects due to vaccination against polio or due to vaccination against MMR, mumps, measles, and rubella. So you have to consider that apparently RNA ain't that bad. The second thing is if we look at the current vaccine, Pfizer, BioNTech, and Moderna, over billions of people have been vaccinated over this last year. There are in fact, there are no clinical trials on any other vaccines ever in which the follow-up has been immediately within a year of billions of cases. So long-term effects, adverse events for RNA vaccines have not proven to be something to be concerned about. In addition to that, there are some extremely rare side effects, if it's thrombosis or if it's myocarditis or if it's allergic response to polyethylene glycol. They do exist, but they are very rare and they are handled, and they're not typically, or they don't have to be life-threatening. And then finally, we want to realize that people talk about adverse events, long-term events and effects, but they are referring to what we know about the long-term effects of disease. Long COVID is not the result of vaccination. We know that shingles is the effect of measles, but not measles vaccines. And there are many cases in which we do know that there is morbidity long-term, but due to the disease, not due to the vaccine. We have to be able to convince the public that this is the case when we talk about vaccines as opposed to disease. Has the writing been on the wall? Now, this is where I think many of us have all screwed up. We anticipated that the minute there is a vaccine, we're going to be protected like in measles, polio, and mumps. But if you look at past experience, cats, dogs, cows, chickens, and pigs all suffer from coronavirus diseases. Many of them have had the opportunity to be vaccinated because of livestock and the commercial aspects of protecting your pigs or whatever. And so under the circumstances, what have we learned from veterinary corona diseases? Well, first of all, we know that natural immunity in these animals from a coronavirus disease wanes. And this is a proven fact for now since the 60s and 70s when these things were studied. The second is that recurrent disease is the norm. We have to realize that each viral family has its own biology. So whereas you can be protected long life from measles and polio, coronavirus doesn't work that way. And when we get infected with coronavirus, immunity wanes and we see recurrent infection in the animals years and years ago and currently with COVID-19. We also know that, to our surprise, I must admit, I was wrong in talking to various people very early on, saying we'll get infected, we'll be protected, we won't get infected, we won't transmit. That is wrong. But if we look at the veterinary vaccinations, we realize that this is the case. And finally, vaccines protect against severe disease, and that we've learned. And ultimately, they require multiple boosts. Now, I'm going to skip this because I promised Adi that I would skip, and we will ask at this point, what is the preparedness for the next pandemic? So I believe that the take-home message is vaccination, and vaccines will play a crucial role in the next epidemic. And this is important. However, there is hesitancy in the population. People are reluctant to get vaccinated, and we say the only way to control the pandemic is to implement vaccination in the general public. But I want to point out there's a difference between vaccine hesitancy and vaccination hesitancy. I've heard many times people tell me, listen, I vaccinate my kids, but I will be damned if I get vaccinated against COVID-19. So there's a reluctance. My concern is because of the messages that people have learned and assume, we're going to see now a reluctance to vaccinate with the general repertoire of vaccines for kids. So what we need is Vaxed. We need to educate for understanding vaccines. We have to do that early. We have to talk to our kids. We have to get our kids to convince their parents. We have to talk to the parents and convince them, maybe, to vaccinate the grandparents. I don't know. But there has to be a dialogue. There has to be education as to what constitutes a vaccine, why is it important, what should we expect, and to be acceptant of the disease we're trying to protect. Thank you very much.
All right, we have time for some questions.
We're making a presumption. All those stuff, personalized mRNA, vaccines, antivirus, people, new technology. Since it's not new, we've already used mRNA, and I was thinking of sugar in fruits versus fish or sugar. There could be some differences because and indicate that millions of doses, billions of doses, don't prove that more adverse reactions, you assume
I didn't hear the question, but I will comment on what I think I understood. So clearly, the statement is that it could be that synthetic pseudouracil containing RNA vaccine is not equal to the use of live attenuated measles RNA vaccine. And to that point, I will say, yes, I agree, it could be. There doesn't seem to be any evidence thus far in dramatic adverse events with the billions of people who have already received the synthetic RNA. That was my second point. So, sure, there may be, and we'll wait and we'll see if there are. And as far as potential other adverse events, again, my understanding of vaccines in general over the past 40 years of research is I would be hard-pressed to find any vaccine that's been used with the old and now the new technologies that have long-term adverse events. There have been cases, such as in polio, in live attenuated, that there's been revertance, and therefore Sabin has been disconnected over salt. So that makes sense. But in a molecule such as synthetic RNA, in a molecule such as in RNA, is that okay? I don't see it happening. But respectfully, I understand the concern, and time will tell us.
All right, we're forced to move on, so thank you, Johnny.